So basically is how do cells adopt a state that is appropriate to their relative position within a developing embryo. And so this is probably the preeminent way in which the embryo, as a ball of cells, knows that these cells here are meant to do something different from these cells here, which is again doing something different from these cells here. So this is important for the coordination in this bag of cells. So as I discussed, I think 2 lectures ago, the developmental programs are generative, they're not descriptive, and all of you kind of got the sense of what this means, and the idea that a developmental program is like origami, that each instruction leads to a particular kind of intermediate form, which leads to the next intermediate form. And all of these different components and different parts at some point start developing slightly independently, so that this developmental program is actually hierarchical, and it's not laid out all at one time. Okay, what does that mean? So it kind of talks about cell interactions, I think, yeah, and this is what we're going to try and talk about today. So development can be mosaic, or it can be regulative. In mosaic development, the embryo doesn't need any further information. When the oocyte is laid and before fertilization, the distribution of determinants that the mother has put in the embryo is enough to make the embryo. And so this is generally the case in arthropods and annelids. So regulative development is the other way around, is kind of almost the opposite. So embryos use interactions to ensure robustness during development, so that external factors do not impact development so much. So the implication here is that the embryo is a blank slate, and particular processes that occur in the early embryo are sufficient to pattern and develop the whole embryo. And this starts implying the importance of cell-cell interactions. I actually think that this distinction between mosaic and regulative development is just a question of timing. There are always cell-cell interactions that occur, even when the mother puts in enough maternal determinants to allow embryonic development. And so I think it's just a question of really perspective. And some of the references that I've just put up in the reading list probably will go into this in a little bit more detail. So one way that patterning can occur is through this famous French flag model. So do you all kind of know this idea here of the French flag model? I mean, I'm sure all of you know what the French flag is. No? Okay.
I studied it before, but I need to brush up.
Sorry, what did you say? Okay.
That I've studied this before, but I need to brush up. I'm not very clear now. I forgot the bits of it.
Okay. Thanks. So basically, what this is telling us is that it talks about something called morphogens, and we'll go into morphogens in a little bit more detail in the next couple of slides. Essentially, a morphogen here is a secreted protein. Could be a growth factor, and this secreted protein is secreted from a source, this point source here, this green cell, and acts across this field of cells. It's able to pattern different cell fates depending on the concentration that those cells receive. So close to the source, the amount of signaling factor that these cells are receiving is high, and so they'll form these blue cells. A little bit further away, they'll form these white cells, and a little bit further away still, they'll form these red cells. And so this this is a kind of French flag hypothesis, which talks about morphogen concentrations acting across a field of cells at different to impart different fates. So this is kind of a very attractive model. It starts getting towards what we talked about in the first or second lecture, this coordinate system that's maybe happening in the embryo. So I think this is kind of clear, yeah?
Yeah.
Okay, great, thank you.
I had a doubt.
Okay,
what
thanks.
are T1 and T2? Is this time dependent? What does that mean?
So they can be time dependent or they're concentration dependent, okay? So hold on to that thought, yeah, we'll come back to that, okay? But so it's going to start getting more detailed as we go on, okay? Okay, but that's a good question, okay? Okay, so this is kind of classically shown in the Spearman-Mangold experiment that we talked about earlier. So here the implication is that the organizer is providing the source for the morphogen. It's able to pattern different structures depending on the distance from the organizer. So close to the organizer, you'll have more lateral components such as muscle, and further away, you'll start patterning more ventral components such as blood or tail regions, okay? And so this very simplistically is a very attractive model. There's various experiments that support this in great detail, but there are some complications. And so the idea of the organizer providing the source for the morphogen is correct in general terms, but let's get a little bit more involved and a little bit more detail on this. So the problem with these experiments and this kind of conception in the first place is the following observations. Yeah, you can cut up the early embryo. So you can cut up the early chick embryo, mice embryos, sea urchin embryos. If you cut up the early chick embryo early enough, all the pieces will form a perfectly fine embryo. And so this kind of tells you that, at least in the chick at these stages, not all, that there isn't a region that's defined as the organizer. So all of the embryo is able to form an organizer. Second observation, you can separate the blastomeres of an 8-cell mouse embryo, and they will all form individual embryos. This is a very known observation. Naturally, I mean, even in us, naturally, sometimes you'll get some event where the two blastomeres will start forming independently, and you'll get identical twins. And so this is a very well-known observation. And finally, you can cut out the organizer from many embryos, and it will still form an embryo. And so definitely in the chick, in the mouse, in frogs, if you do it early enough, if you remove the organizer from the embryo, an embryo will still form. Moreover, the gradient scale. So you can cut Xenopus embryos at least, and of course the chick embryo and the mouse embryos, and you'll still get an embryo. It'll be maybe smaller, but it will form just fine. And so what you're seeing is the embryo scaling according to size. So here, this is another very famous experiment done by Spearman. What he did was cut the embryo in half. So he took the embryo when it's at the 2-cell stage and cut it. And actually, he used the hair of his newborn daughter to make a loop and tie around these 2 blastomeres and separate them, so that now he has 2 blastomeres independently developing. And what you get are 2 smaller embryos that look perfectly fine. Okay. So the question then becomes is, well, what's happening to the gradient to give us this fine looking embryo? You can also do this at 1 division later. And so as you remember, the maybe I didn't tell you. So in Xenopus cleavage, the first division separates right and left halves of the embryo. The second cleavage, so you now have 4 cells, separates the dorsal and ventral halves of the embryo. And so what Spemann had done is separate the right and left half of the embryo. So you're still keeping a dorsal piece, half of the dorsal organizer in that 1 blastomere. You can do this 1 division later, so you can take ventral and dorsal halves and ask what they give rise to. And this was done in this experiment here. If you isolate the dorsal half and the ventral half only forms ventral structures, okay? But the dorsal half now forms a perfectly fine embryo. So something is happening that enables at least part of this gradient to be reconstituted in a very robust and size-independent fashion. And so what we're going to talk about for the majority of this lecture is kind of what these mechanisms could be. So do we have any questions up until this point? Questions on frogs, chickens, mice, flies, fish, worms? Fantastic.
Raj, I just the slide that you're showing here, this is of which embryo?
So sorry, yeah, this is a frog embryo, a Xenopus embryo.
Okay, okay, thank you.
Yeah. Fantastic. Okay, so the same kind of experiments can be done in zebrafish as well. And so here, what's happened here is that around 30% of the embryo has been removed by just simple extirpation, they've called it, but it's just sucking up cells. And what you see in an extirpated embryo is perfectly normal development. And so what you're looking at here is pattern of the neural tube here and here, showing very nice, absolutely normal development. Okay.
Hi, I had a question.
Hi, Meghana. Yeah.
Yeah, so if we cut the embryo in such a way so that the part which I expect to form the head and the part which I expect to form the tail, they are separated, what happens then?
So this was done here in this Xenopus experiment, and so here the tail half will form tail-like tissue only. Yeah, it doesn't form head-like tissue, but the head half will form head and tail-like tissue. Okay.
Yeah, okay.
Yeah. So this is I should say that all of these are not general. So this is kind of something you see in amphibians. In chick, depending on the time you do these experiments, you can reconstitute a whole embryo if you cut dorsal and ventral halves. As you get later and later in development, you'll start seeing more and more restriction to either a ventral or a ventral fate. The ability of something that contains the organizer to also make then a ventral fate as well, or a tail fate, is not so restricted. And so that ability continues for quite a long time. Okay.
Yeah. Okay, thanks.
Okay. So fish do it as well. Okay, and so how is this happening? And so what this kind of suggests to us is that the sources of signals, so the organizer, is not necessarily fixed. And so even the organizer itself is under some sort of dynamic regulation, which is what we're going to talk about now. Okay, and what we need to do to talk about it is just to remind you of this reaction, the Belousov-Zabotinsky reaction. Okay, and so what this reaction is, and I'll explain this in much more detail now, is a reaction where bromine and bromate are being produced, and it's kind of an oscillating reaction. There's kind of the need for catalysis. It's, you know, the general scheme is shown here, but what you're getting is that at some point bromine, which is of course brown, is depleted and forms the bromate, but then the bromate again will degrade and form bromine, and this cycle will just keep continuing. Okay, and you get these beautiful patterns. Yeah, so you get these swirls here that are formed in a kind of so this is not so this is one of the first chemical reactions where it seemed like heat didn't really play a role. Okay, and so this is kind of this very beautiful natural oscillation happening in this just simple chemical reaction. This kind of scheme was more formalized by Alan Turing, and so probably many of you know Alan Turing as a kind of computational mathematical genius, played by Benedict Cumberbatch in the Imitation Game. Imitation Game, is that right?
Yeah.
Yeah. Okay.
Yes.
Yeah. Okay. So I'm sure you've all seen the Imitation Game. You all know about Alan Turing's life, and he probably made wrote one of the most beautiful papers on the theory of developmental biology and the theory of signaling, which is now called the Turing model. Okay, and so this is based on reaction diffusion, and we'll talk about this in the next slides. But what he essentially said was that you have an activator which will turn on its own expression, but it will also turn on the expression of its inhibitor, and its inhibitor feeds back to repress the activator, and in this way you start getting various patterns which can be represented here. Okay, and so here this beautiful review from Shigeru Kondo talks about these different abilities. And so if you have one morphogen, and just the simple morphogen that we're talking about in the French flag model, you can get kind of 1D patterning, so one-dimensional patterning, either horizontal or vertical. If you now have two morphogens laid on top of each other, you can start getting slightly more complicated patterns, but not very complicated. But now if you start invoking this reaction diffusion mechanism where you not only have two morphogens, but the morphogens talk to each other, and so they're dynamically regulating each other, you can start generating very beautiful patterns. Here it's spots and stripes, or a labyrinth kind of pattern, and all of this depends on the diffusion abilities of the activator and the inhibitor, and the repressive ability of the inhibitor, the activating ability of the activator. And so this is a very elegant way of looking at signaling interactions. Any questions here? Okay. So these kind of interactions are used in various developmental schemes as well. Here, this interaction between Wnt signaling and the names of the signaling pathways aren't important right now. Okay, I can put up a review on signaling pathways if you want. I know many of you are not biochemically inclined and don't really want to know about signaling pathways, so I left out those kind of details. But Wnt is a secreted factor. DKK is its secreted inhibitor, and this kind of interaction is known to be involved in things like hair follicle spacing, hydroregeneration, lung branching. These two signaling factors seem to be operational when you have left-right asymmetry, and so on. We'll talk a little bit more about these kind of interactions in the next few slides, okay? But all of these interactions are kind of predicted by this reaction diffusion equation, which Alan Turing formalized in his classic work, The Chemical Basis of Morphogenesis. Okay, so what we're basically seeing is an activator which activates itself and also leads to the expression of an inhibitor, and the inhibitor, of course, is acting back on the activator. And so what will happen, and so this is a very nice review from the late Hans Meinhardt, and what he's modeled is even when the activator starts off at a baseline concentration, small stochastic mechanisms or slight instabilities may cause the activator to be expressed. This expression, of course, leads to the expression of the inhibitor as well, but just after some time. So the activator is able to activate itself. Here we're seeing the start of this instability. This is magnified so that you now have a sharpened gradient, okay? So this is, I think, all well and good. You also can see that there's a gradient of the inhibitor as well, a much shallower gradient of the inhibitor. So these are sea urchin embryos, and what you're looking at is the expression of one of the a molecule that is acting as the activator, and what you're seeing here is after cutting. So here, down here in EFGH, after you cut a sea urchin embryo in half, what you're seeing is now this gradient has been cut in half. So you're seeing the dorsal side with the steep gradient and the inhibitor gradient quite high here. But on the cut in the ventral half, you're seeing a much shallower gradient. This shallower gradient collapses, but then instabilities in the gradient allow it to then reform. And so this gradient, by some instability, which is still not really well characterized, will start to, the activator will activate itself, it will form the expression of the inhibitor as well. Sorry, it will induce the expression of the inhibitor. And we now are left with a situation where we now have two halves of the embryo that have two very nice gradients forming. Okay, I think I did a terrible job of explaining this. So if you have any questions, please ask them.
Raj, could you explain once again what happens when we go from the From E to F?
E to F. From here to here?
Yeah.
Yeah, so what's happening is, so E, I think, so the left-hand side is fairly clear, I think, yeah. And so this is, for example, the dorsal half of the embryo. Its gradient is basically maintained and sharpened. Okay, so let's look at the right-hand side. Let's call it the putative tail half of the embryo. Essentially, because the expression of the inhibitor, what we're left with is now the activator was essentially at 0 in that tail half. The inhibitor was kind of high because of the action of activator from this region, from the left half. Now that the activator is low, the inhibitor, which is depending on the function of the activator, is now brought to 0 as well, essentially. Now, between E and F, we have some process that I think we don't fully understand. And so these are all simulations that Meinhardt has done. But I think there is some instability in this region, which causes the activator to be reinitiated. The reinitiation of the activator causes a high induction of the inhibitor, which kind of brings down the gradient a little bit, so that we now have this final form here in H. Is that kind of clear?
I think I have a better idea if I look up the review.
Okay. Yeah. So the review also doesn't explain this so clearly. And so
Raj, could you explain the could you explain the figures, please? I could not understand. I mean, what is being indicated in the figures?
Okay. So what we're seeing here in A is the hypothetical reaction diffusion mechanism. Okay, and so in A, so what we see is the activator turns on. An activator turns on its own expression. So an activator is required for an activator. But the activator also turns on the expression of an inhibitor. Okay. The inhibitor, of course, feeds back and represses the expression or the function of the activator. Okay. In these B figures here, what you're seeing is the formation of the activator gradient. And so initially everything is at a baseline, okay? And then there's some instability that takes place, which causes maybe the expression of the activator to be initiated in some portion of the cells. And so now that the activator is initiated, of course, there's a positive feedback loop on activator expression, and so activator goes up. Okay. The activator also causes the expression of the inhibitor, this red line here, which increases inhibitor expression as well. And so in this, what you're seeing here are particular sets of values for the diffusibility of the activator and inhibitor, how well the inhibitor is, how good the inhibitor is at inhibiting the activator. And here you're seeing this particular gradient forming. Okay, is that okay so far?
Yes, and the figures below, I mean the CD, those slide pictures. So
Okay.
are they related to the gradients?
Yeah. Sorry, I didn't. What you're looking at, C and D, is so C is the formation of and so this gene is goosecoid, and it's a dorsal marker, and you're looking at sea urchins. And what you're seeing here is the normal expression of goosecoid expressed over time in a sea urchin embryo. Okay. D is a half sea urchin embryo. So it's been cut, and the gradient has reformed, and that's what you're looking at here. Okay. And then EFGH is modeling what's happening to the reconstitution of these gradients in the cut halves and the two halves, basically. Okay.
All right. Thank you very much.
Okay. Okay. So actually, this is the simple one. Okay. The slightly more complicated version is what actually happens in Xenopus and in mouse. Okay. And so in this generalized model for axis formation, so for basically dorsal ventral formation is what you're seeing here. What Meinhardt has done as well is kind of put in an argument for an anterior posterior organizer as well as a dorsal ventral organizer, which I don't really want to go into at the moment. But suffice it to say that BMP, which is another signaling molecule, is found on the ventral side of the embryo. It causes, it inhibits the expression of caudin, and caudin is an inhibitor of BMP. Okay, caudin also works on this other factor called ADMP, which is also an inhibitor of a dorsalizing signal. And so what caudin, the expression of caudin is regulated by upstream molecules, and these include nodal and other members of this family, which I'll go into in the next couple of slides. Okay, but the same kind of scheme of dorsal and ventral counterbalance. So the dorsal structure, the dorsal organizer inhibiting, making its own inhibitor and activating its own activator works even in Xenopus and in mouse as well. Okay, and this is one way that gradients are stabilized in these vertebrates. And so I'll show this better in this slide. And so this is kind of the essential scheme for how dorsal ventral patterning is happening in Xenopus, and it is similar for mouse as well. Some of the names are a little bit different. So essentially, what's happening is you're having a system of activators and inhibitors. The gene crescent is working to inhibit this molecule called tolloid. Tolloid is a protease which cleaves caudin. Caudin normally inhibits BMP, but because it's cleaved by tolloid, it allows BMP to be repressed in this region, enabling dorsal formation. ADMP is also an inhibitor of dorsalizing activity, but it's formed in the dorsal region, but it feeds back onto BMP in the ventral region, activating BMP expression. You have kind of these fairly complex networks of activating and inhibiting compounds, which all they're doing is stabilizing dorsal and ventral formation. And so in this way, what you can see is that when you cut the embryo in half, this complex regulation is reformed in these new halves using the same kind of thinking shown here by Meinhardt, enabling gradients to be reformed. Okay, so this is this is in dorsal ventral patterning in Xenopus. What you're looking at here from this Almudo Castillo paper is left-right patterning, and left-right patterning is very similar. It uses this balance between two factors called nodal and lefty, which ensure that you have differences between left and right of the embryo. Okay, and I'm gonna, I mean, you can see these papers for more details. Okay, questions here?
Raj, could you explain why it's called flux over there, or what the flux is? The red arrows.
Yeah, so the flux is oscillations basically of chordin BMP or ADMP and BMP, and so these oscillations are set up just because of the normal feedback mechanisms of this group of proteins.
Okay.
So these are always being produced as well, but their activities are being repressed in kind of an oscillating fashion. Okay. Okay. So similar kinds of these kind of networks are operating in different places as well. So when you will start talking about fly development with Tina, the same kind of interactions between caudal and bicoid in patterning the tail and the head of the fly are operating, and these are kind of manifest in the expression of these gap genes here. Similarly, in the patterning of the neural tube, which Sonia and Bhavna will talk about in a few weeks, you get similar opposing gradients with opposing feedbacks, which are patterning very intimately the dorsal ventral parts of the neural tube. So, so let's ask, so what's the consequence of all of these signals? And so signals are integrated at enhancers. And so all of you know what enhancers are. Is that correct? Can you hear me?
Yes.
Yeah.
So enhancers are basically sequences on the sequences of genetic material which are usually occur before the gene, and these are sites for binding of activators which can activate the transcription of the genes.
Yeah, yeah. So that's a good definition. So definitely they're basically control elements of genes, okay, and they're in the nucleus of a gene. They're bound by particular transcription factors which will drive the expression of an effector in those target cells. And so this is how kind of the primary consequence of receiving particular levels of signaling molecules. You'll have particular the ability to activate particular genes that then is registered in the nucleus. So what you're seeing here again is a simple gradient, either a blastoderm or a neural tube here, with particular activation. An enhancer can be repressed, it can be constitutively active, or it can be at particular time points affected by a morphogen. And so all of these three elements within an enhancer will cause the expression of a downstream gene. And these genes will be ones that will control cell behavior, for example, enable a junction to remodel or something along those lines. So just as there are kind of intimate networks of signaling molecules upstream of these transcription factors themselves have very complicated dynamics as well that ensure proper pattern. And so going to going to, so what does this mean? And this is a kind of addressing Megna's question earlier about time. So of course, one feature of a morphogen gradient, and so here now we're not really thinking about that complicated structure of many proteins acting on each other. We're just looking at the readout of a particular morphogen gradient. It's increasing in concentration along this green line here. We now have to kind of integrate this idea of time into this morphogen gradient as well. And so, of course, the morphogen gradient is acting over some degree of time. What the final readout of this morphogen gradient is down here. So you have blocks of blue, orange, cyan, and is this red? Teal? No, ochre, brown. This color here forming. Okay, and so what Meinhardt has modeled is what goes on with what goes on in the nucleus in response to this gradient. And so what you're seeing is that at a particular time you have this first gene which is sensitive to the least concentration of the morphogen coming up. Okay, a little bit later you start seeing other genes coming up, and so these are depend on higher amounts of the morphogen, but there's networks within these within the enhancers of these genes which exclude each other from the cell. So a cell that's expressing a blue gene will not be the same as a cell that expresses an orange gene, will not be the same as a cell that expresses a cyan gene, and so on. And so in this way, you start getting sharp borders between these different territories, okay? And so it's not just dependent on the upstream signals here, okay? So the morphogen gradient is fairly continuous, but what you're seeing in the nuclear response is very discontinuous, and that sorry, the reason for this discontinuous kind of
discontinuity.
Thank you. Say that again.
Discontinuity.
Yeah, I don't know why I can't say discontinuity. Yeah, it's yeah. So thank you. So the reason for this discontinuity is because of the way the enhancers are wired in the nucleus. And so, like I told you, enhancers can have repressors, activators, and kind of effectors, which are responses to the morphogen. But remember, so it could be that the red gene acts as a repressor for the blue gene, is the easiest way to think of it. The cyan gene may act as a repressor for the red gene, and so on. Or it could be slightly more removed from that. The red gene could induce the expression of a repressor for the blue gene, and so on. Okay, and so in this way, you're starting to see how this continuous morphogen gradient can induce these kind of specific cell fates with sharp boundaries downstream. Okay.
I had a question.
Yeah.
Since it is in like morphogen is being exposed in a continuous manner, so what if the morphogen suddenly goes out of the picture? Will this still be persistent, or all of them will collapse and then it'll all come down to zero?
Oh, that's a great question. So there's kind of this time dependence on this gene expression as well. Okay, and so there's probably a time at which if suddenly you took the morphogen away, then everything would collapse to a baseline. But at some point, these kind of different fates become maybe fixed, yeah? And so you can see here that there's some fixation as you get to more distinct discontinuities. Yeah. And so it could be that by this point here, there's less dependence on this morphogen being there. Okay.
Okay.
Yeah. So probably the right answer is it depends, and it depends on the structure and the system that you're looking at. So this kind of concept is certainly true in neural tube patterning, and I'm sure you'll talk about this when Sonia and Bhavna will talk. So the neural tube is also a result of opposing gradients that interact, gradients of sonic hedgehog and BMP. And you can see very clearly from this paper and a very, very nice paper from James Briscoe's lab that you have the effectors of these signaling molecules, of these morphogens, expressed fairly in a very continuous fashion, and then you start seeing discontinuities that fix particular gene expression domains within the neural tube, and those are related to particular populations of motor neurons and sensory neurons within the neural tube. And in this way, you start getting these kind of this integration at the enhancers of all the signaling factors, and so like and you'll see this in Drosophila embryos in dorsal ventral patterning. You'll see that morphogens will activate various combinations of enhancers, which will drive various patterns of gene expression, and remember, these effector genes are leading to changes in cell behavior, changes in cell potential, which are driving development. Okay, and this you're seeing here is a neural tube in E. The neural tube has a gradient within the nuclei of these particular factors. These are GLI, the GLI transcription factor, either the repressor or the activator form. It's on a background of SOX2 with these particular effector, these particular activators expressed kind of basally in these different domains in the neural tube. And so what you're seeing here again is because of this kind of combinatorial action within the nucleus, which is only partially dependent on the signaling factors. So GLI is dependent on the dorsal ventral patterning in the neural tube. You start seeing very specific gene activity within subdomains within the neural tube, which will drive gene pattern within the neural tube, which will give rise to particular populations of neurons. Okay. Okay. So as well as the neural tube, also we see this in the limb bud, in the limb. The limb is also subject to signaling. You'll know much more about this in the last couple of slides in December, last couple of lectures in December. But I just want to come to the point of here, looking at the hand of the limb. So one thing that's fascinating is how the digits in the limb form, okay? And all of these blue, what you're looking at here in blue are the cartilage elements. So these are condensations that will form cartilage. In red is kind of the ossifying bone. And so what we're going to think about a little bit is how the cartilage elements are formed. And we're going to also relate that to reaction diffusion mechanisms as well. So this very beautiful review from Jeremy Green and James Sharp, talks about these Turing patterns, and what they've suggested is that if you can lay on these kind of oscillating wave-like patterns on a positional information, so a different morphogen gradient that's causing a French flag type of approach, you can start seeing very beautiful patterns starting to emerge. And so what they've looked at here is they've tried to model these kind of digit formations in the hand. What they've come down to, and this is a very beautiful paper from James Sharp's lab, and what they've come down to is this kind of network here, okay? And what they've essentially shown is that this this transcription factor SOX9 is important for cartilage formation, and so that's known. SOX9 is inhibited by this signaling factor Wnt, but activated by this signaling factor BMP. And this kind of complicated, not so complicated, this kind of wiring diagram is sufficient to start forming these stripes of SOX9 that will prefigure the digit formation in the hand. Okay, and so this, the kind of cool thing about this is that laid on top of this is a morphogen gradient that's going across the hand that lets you then distinguish where your thumb is and where your little finger is as well. Okay. Cool. So this idea of combinatorial signals, all kind of working together to give you not just repeating digits, so 5 digits, but also then pattern across the digits. So thumb to little finger is very powerful. So like I said, we've gone through this. So this is a way to fix position after signaling. But what happens next? Okay, so when we fix this, when we start getting these 4 domains of blue, red, cyan, and brown gene expression domains, what will happen next? And so, of course, you'll start seeing cell behaviors, but also these borders tend to be very important. And so what you're seeing in this next half of the slide is what's happening in the development of the fly limbs, okay? And so the fly limb seems to form at particular boundaries between different gene expression domains, okay? And in fact, the point of outgrowth of the limb is formed here between this anterior and posterior stripe of the fly, and just here between this medial and lateral parts of the fly larvae. And so these kind of borders, these boundaries, can act as then organizers of a secondary embryonic field. Okay, and so now, because they're specified, because they're now at boundaries, they're seeing 2 different signals, they're experiencing 2 different factors, and in many cases they start doing something very different. This kind of is I don't want to go into this figure too much, because Tina will talk about this as well, but again, it's in this Meinhardt review. It's a very attractive idea of thinking of these kind of boundaries. So either here, these very concrete boundaries, which are boundaries within alternating segments in the larvae, but here also these boundaries in this computer model are now different. So this region here is different from all the others. Okay, and these now act as new embryonic fields. And so what does embryonic field mean? We go back to our origami picture. So embryonic fields are now, for example, once you fold the origami enough, it's a region of the origami which can do something different from this region. So it can have its own signaling profile, its own induction system. The cells can behave almost independently from the embryonic fields around it. Of course, it's still integrated into the body, but in this way, because you now have secondary embryonic fields, you can start exploring many more different phenotypes. And so you can imagine that if one embryonic field, for example, expresses a particular transcription factor, as well as being these particular boundaries, that embryonic field, rather than forming a limb, will start to form a wing. Okay. So, any questions up until now? Okay. So, all of this is somewhat dependent on these reaction diffusion mechanisms. So, of course, reaction diffusion mechanisms operate very visibly. So in 2 dimensions, you can see here, this is a mutant mouse where, it's a genetically engineered mouse, again from Shigeru Kondo's lab. And so what they've done is to put a kind of stop on the tyrosinase gene. And so the tyrosinase is important for pigmentation in the mouse, and you can start seeing these different pigmentation patterns starting to appear in the mouse. And these pigmentation patterns seem to be reminiscent of particular reaction diffusion mechanisms. And so here again, this is the BZ reaction overlaid with the mouse, showing these patterns of pigmentation, which are possible when just by playing with the chemistry of tyrosinase and its degradation. And again, I will direct you to this paper here for more details. Okay. Another place where this kind of reaction diffusion mechanism happens, and so someone asked me in the first question, the first lecture, about single cell organisms differentiating. So this is a cyanobacter. It has 2 types of cells that form in these filaments. A cell that expresses HETR, which fixes nitrogen, and these non-fixing cells that surround it. These nitrogen fixing cells express HETR. The HETR will turn on the expression of HET-N in adjacent cells, and HET-N then restricts HETR. Okay, and the same with this other transcription factor, PAT-S, which has a similar relationship with HETR. Okay. So what you start seeing then is this kind of periodicity of these nitrogen fixing cells in cyanobacter along this filament. And this is all, these are of course unicellular colonies. They're forming a filament, and they're showing this kind of pattern. Here, this is HETR GFP expression, and you can see that you have these regions of high HETR expression and then low intermediate HETR expression, which are these non-nitrogen fixing cyanobacter cells. Similarly, in the Drosophila sense organs, so fly sense organs, insect sense organs, also have a similar scheme. The red cell here is a putative sensory organ in the in Drosophila, it activates itself, but also inhibits neighboring genes to adopt the same fate as itself. And this is dependent on a particular signaling system, which I'll introduce in the next couple of slides. But any questions up until this point? Okay. Cool.
Raj?
Yeah.
I was wondering about the embryonic fields. So theoretically, can they be, say if you take an embryonic field outside of the organism, can they be grown in a dish into the relevant organ? I mean, or whatever unit it is forming?
Yeah. It can be. It can be. So definitely in some animals that's really easy to do. In some embryos that's really easy to do. In Xenopus that's crazy easy to do. You can take and you can start seeing the appearances of these embryonic fields as well. So one thing you can do is cut out the neural tube of a frog embryo and culture it in isolation. And you can see that at a particular stage, it will form structures that may be equivalent to a forebrain. So you can start seeing eyes and a telencephalon forming in these structures. But then at later stages, the same piece of tissue then starts forming midbrain kind of structures as well. Okay, so yeah, you can definitely do that. I know you can do that in frogs. Okay. You can do that in chicken as well, chicken embryos, and it just depends on how well you're able to culture these fragments. So many embryos are more resistant to being cultured ex vivo, but some are very easy. Okay.
Yeah, but you said that the signaling profile is sort of independent while it's still integrated to the rest of the organism.
Yeah.
If the signaling profile is independent of, say, neighboring embryonic fields, then it sounds like it should be able to be grown.
Yeah. I think it just becomes a question of how easy it is to, it just becomes a technical question, how easy this is to grow. Yeah.
Thank you.
Okay. So again, this is again the SOX9 Wnt BMP pathway in the limb bud, another example of these kind of reaction diffusion mechanisms that start to cause digits or cartilage condensations to start to appear in this very patterned fashion in the hand plate of a mouse embryo here. Okay. So the last thing I want to talk about is somatogenesis. Does everyone know what somites are?
They're blocks of mesoderm that are formed in the embryo, which later differentiate to form various structures.
Yes. Can you remember what those various structures are?
The dermatome, the sclerotome, myotome.
Okay. Yeah. So you know a little bit more than I do. I was just going to go for vertebrae. Okay. So one of the main things they form are the vertebrae. But as Shubha says, they're blocks of mesoderm in the tail of the embryo, and they will form vertebrae and various muscle blocks as well. And you can see you can see them forming here. Okay, and so this is a chicken embryo being cultured, and at some point you can start seeing the somites starting to form. First somite will start to form around here. There you can see it. Then the next one, then the next one, and so on. Okay. So one question always has been how these somites form, and if you notice, they're very periodic. In in chickens, one somite is formed every 6 hours or so. In mice, the periodicity is a little bit longer. So one somite takes about 150. It's about 3 hours. In human, the periodicity is much longer. Yeah, we're starting to look at about 4 hours, I think, 4 to 6 hours in humans. Okay, so somites have a periodicity. They're formed in a periodic fashion, and so what I'll try and do is explain the final kind of signaling process, and try and integrate everything that I've told you about by thinking about somite formation and their periodicity. So it's thought that, well, it's fairly well accepted that somites form, so how do you get this periodicity? And so one way that you could do it is if you had oscillations going on in the mesodermal lineage that will give rise eventually to the somites, but also so here you're seeing these oscillations happening, but also you're having a gradient, okay? And so as long as these oscillations are happening within the confines of here this FGF gradient, FGF and Wnt gradient, they don't do anything. But once the oscillations happen outside of the FGF gradient, you'll start seeing pinching off of somites, okay? And so this is called the wave clock front model for somatogenesis. It's a very beautiful and attractive model, but one question has always been how this periodicity, how these oscillations in the future somites could take place. And I want to bring to your attention another BZ reaction, another oscillating reaction called the Briggs-Rauscher reaction. And so this is now in solution, okay, and it's a mixed solution. It's the catalysis of iodine and iodite with starch as an indicator. Adding hydrogen peroxide here. I think the iodine solution there with starch, okay? And so you see it goes, the iodine is there, causes this purple reaction, which is then cleared as the iodite goes down into iodine as well, okay? So again, this is a simple oscillating reaction that is occurring because one of the products of this, of the blue, so the reaction that causes the indication to blue iodate is being broken down to iodine, okay? But then the iodine is now being catalyzed to iodate. Okay. So this kind of, so we can see oscillations happening, even chemically. Can we see them happening in the embryo, in the future presumptive mesoderm? So what you're looking at here is a reporter, and I'll tell you what the reporter is soon. But the reporter, you can see, is showing very beautiful oscillations. What you're looking at is the tail of a mouse embryo, showing this reporter gene expression that glows at particular time points. And you're seeing very beautifully these oscillations traveling up the tail, meeting a wavefront, and pinching off into somites. I'll show this again because it's really beautiful. Okay. Cool. So this kind of scheme, this reporter doesn't even need an intact tail. And so what's happened here is that the tail of the mouse has been dissociated. The cells spread onto a dish, and now the dish is being imaged. The cells are being imaged, and the cells have this reporter that's showing this oscillatory behavior. And let's see it here. And you can see again, there's oscillations of the reporter happening even in these dissociated cells. And so there seems to be some sort of intrinsic behavior. You're seeing the oscillations now breaking down as we go a little bit further. But this behavior seems to be intrinsic to these cells. And this behavior is dependent on the notch pathway. I'm going to let you read about the notch pathway, but for the sake of this for this lecture, all you need to know is that these two molecules, so delta or delta-like 1 is on a signaling cell, and the notch receptor is on the receiving cell. This interaction between delta and notch causes notch to be cleaved, the intracellular domain of notch to be cleaved. The intracellular domain of notch is now able to act as a transcription activator in combination with proteins that are sitting on the DNA, and this then causes the transcription of this group of genes, lunatic fringe, HES7, and Notch1. HES7, so Notch1 of course is the receptor again. Lunatic fringe glycosylates the notch receptor to block it from signaling to delta. But also HES7 then acts as an inhibitor to the upstream transcription factor, blocking HES7 expression. Okay, so you can see now that we have kind of the elements of a reaction diffusion, mechanism, or a kind of feedback loop where notch is activating itself, but also inhibiting itself as well. Okay, and so this plays out in a very interesting way, as shown very recently. So it seems that the oscillations are very much dependent on this HES7 gene, okay, which is induced by notch signaling. And so this has been shown classically by Julian Lewis and Nigel Holder here. I'm not going to talk about it too much, but I'm just going to show some of the consequences of this. As I said here, this is the pathway in somatogenesis. So notch activates HES7, which blocks HES7 activation essentially, okay? You have all of these other components feeding into this pathway, FGF, for example. But essentially, I want you to focus on this notch HES7 regulatory network. So if you remember, I told you I showed you this reporter, and so what this reporter is the HES7 enhancer hooked up to GFP. Okay, and so what you're looking at is activity of HES7.
Raj?
Yep.
What did you mean when you said that the break in that is intrinsic to the cells?
So what I meant was that we'll go back. What I meant was that it seemed like the cells were, so all the cells were had this this HES7 reporter in there here, but the reporter is inside the cell. It's in the nucleus of the cell. So this is a transgenic mouse, and even when the mouse is or the limb bud is dissociated, you still see this kind of oscillating expression. Okay, and so here, I mean the cells do talk to other cells, but there's a transgene which is inside, which is incorporated into the genome of the mouse, the mouse embryo. Okay, so that's kind of what I meant. That it was an intrinsic property of the mouse. Okay, and that kind of becomes a little bit clearer when we think about this last second from last slide. Okay, as I mentioned, the somatogenesis, the periodicity of the mouse and human are different. Yeah, so the mouse will form its somites much more quickly than a human forms its somites, and so this is work done in actually differentiated ES cells or IPS cells. So you can differentiate stem cells to form what's called pre-Semitic mesoderm. This pre-Semitic mesoderm is expressing this Hes7 reporter. And you can see this Hes7 reporter now flashing at various periods. And you can measure that periodicity and understand what the periodicity of this somatogenic clock is. In the same way, you can differentiate human stem cells to form pre-Semitic mesoderm, and then again measure what the periodicity is in these cells, and you find that the periodicity in these cells is longer. Okay, so this kind of lets us then ask what the difference between human and mouse is. So why is the same reporter, this Hes7 reporter, acting differently in mouse or human pre-Semitic mesoderm? Before I go on, does anyone want to hazard a guess? I'll bring back this slide. Okay. Does anyone want to guess at where the difference would lie? Okay. So it seems that periodicity.
Could it
Yeah.
depend on the rate of protein production, RNA degradation, and all of that? Is that different?
Yeah, fantastic. Yeah. It absolutely is. So it really is dependent on this rate. Okay, and so here this experiment is to so what they've looked at is putting in mouse Hes7 into the human pre-Semitic mesoderm, mouse Hes7 into human pre-Semitic mesoderm or mouse pre-Semitic mesoderm, and showing that the gene itself per se doesn't really make a difference. But when they start playing around with whether it has an intron or not, so introns are a way of slowing down the formation of mRNA, at least, yeah, they start seeing a change in the Semitic clock. And what experiment is not shown in this paper, which the last author of this study, Miki Abisua, showed me, is that it seems like there are kind of degradation pathways that are specific to the mouse and specific to the human, which are controlling this periodicity. And so it seems like, as you say, the degradation of the protein at this last step is important for controlling the periodicity of this clock. Okay, and so I think I put this paper up on the reading list. Have a look through this, and there's another paper that talks about the clock, the differentiation clock for motor neurons in human and mouse ES cells as well, as a way of understanding what drives kind of species-specific timing for differentiation. Okay. So, like I said, this is very much dependent again on this pathway that goes from Notch signaling on extracellularly, turns on Hes7, Hes7 RNA and protein degradation rates determine the oscillations of Hes7, which determine when a somite appears and how big a somite is as well. Okay. So I think that's it. So that's it. Hello, can you hear me?
Yeah.
Yes,
Yeah.
Raj.
Yeah, I suddenly lost the slide. Is all. Okay, so that's all. So we finished the lecture today. If you have any questions, so please ask.
I have a question.
Yep.
So you stated that it was the intron presence that is the reason for slowing the translation, and hence the difference between the mouse and the human expression pattern.
Yeah.
So can we correlate this with mRNA production, like the way the mRNA processing is taking place?
Yeah. So the presence of the intron was one way that they had devised to slow down mRNA production or mRNA translation. But they also suggested that there are different mechanisms as well. So when the protein is produced, there are its ability to be degraded is very species specific as well. So the mouse protein and the human protein will be degraded at different rates in both of these different animals.
Is there any reason for such specificity?
There's the question.
Okay.
I'm sure there is. Yeah, and I'm sure that things like the speed of any of these degradation pathways that you can think of could be different. But yeah, I mean the lab which produced that study is trying to expand the study to think about elephants as well and shrews. So elephants, of course, have a 2-year gestation period. Mice have a 20-day gestation period. And so thinking about what the difference is, why those differences are hardwired into different embryos is kind of where they're going with that study. But the oscillation work in the somites suggests that these kind of networks where the activator activates itself but also inhibits itself as well are going to be important in stabilizing, counterintuitively stabilizing particular patterns of signaling morphogen gradients, of gene expression patterns and things like that. Okay.
Okay, thank you.
Raj, I was just wondering if we can control the periodicity, then hypothetically, would we be able to reduce the gestation period of certain organisms?
I guess so. I mean, the periodicity isn't related to gestation period, but I think it is related. So if you can do it on a global scale, then maybe. But then I'd imagine that the mother would have particular mechanisms that she knows. If you're a mouse, she knows that 21 days. This embryo needs to get out. Yeah.
Okay.
I wonder if that's happening as well.
Yeah.
Yeah.
Okay.
Yeah.
And if we reduce the absolute time, won't that affect developmental time?
Yeah. Well, it may not. And so it could be that if you're doing this globally, that the whole of development kind of either slows down or speeds up accordingly. Yeah. I mean, these are kind of almost near philosophical questions. There's a very nice review from Ibitua and Briscoe, which I'll put up on the Moodle page after this lecture, that you can read as well. Okay, and that maybe goes into all of this idea of developmental time in better detail than I would. Okay.
Yeah, thank you.
Okay. All right. So as I said, on Friday, we'll have a quick lecture, but then a longer discussion on some of these points. Okay. If you're clear, if you're unclear about some things or have some doubts about some things, then use that as a way of just quizzing me and or correcting me if you want, so that you get more clarity. Okay. Okay. So with that, I'll let you go. So thank you very much, everyone. Have a lovely day.
Thank you, Raj.
Thanks.
Thank you.
Thank
Thank
you.
you.
Thank you.
Thank you. Thank you.
Thank you.
Thanks.